Hello biology students! Today we are going to be working on our DNA foldable, talking about the parts of DNA, its structure, and replication. In 2019, this is part of your packet, and you will find it starting on page 4. You will also need from your teacher the worksheet that has 4 columns. We are going to quickly look over the background for some key vocab words we've been learning through our flipped notes, and then we're going to talk about how to actually build our paper model. All right, let's begin. So in the background, we can see a couple of important words like DNA. We can see and remember that DNA is in the shape of a twisted ladder, a double helix. We can learn and remember that DNA, if we think about that ladder that is this shape, like a ladder, the sides of the ladder, okay, the sides are made up of sugars and phosphates that are alternating. And on the inside parts of the ladder, we call those things nitrogen bases. Those are our A, T, C, and G letters. There, again, are those 4 types. T stood for thymine, A for adenine, C for cytosine, and G for guanine. Whew, thank goodness we have those shorter ways of saying it. And we even know which letters go with what, called the complementary bases. T can only link with A, C can only link with G. Remember our car in garage, apples in tree saying to help us remember the complementary base pairing rules. Okay, we know that a part of DNA is called a gene. It's a section of the DNA ladder, maybe 100 A's, T's, C's, and G's long. And the order of those A's, T's, and G's is the genetic message. The order of mine is different than the order of yours. So now we're going to have our piece of paper. It's actually not construction paper. And we are going to fold it. So at this point, we're going to put our packet kind of up and off to the side. And please follow along and fold with me. So now let's get our sheet. And we're going to fold it hot dog style. So we want the letter A to go with the letter D. So we're going to fold it so that they touch. Oh, so cute. They're touching and giving each other a hug. Now this is not going to fold up perfectly. I'm definitely not a perfect person. We're going to unfold it. So now there was this crease down the middle between B and C. Okay. We're then going to fold it again back in between where A and B are going to touch now. This part's a little hard, so I'm going to do it twice. So now my crease is messy. I have a fold between A and B. I'm going to also fold again the other way, so there's a crease between C and D. Notice that I'm not cutting anything, I'm just folding. No scissors required for this task. So now there is a crease going between each of them. But if I folded it perfectly, I should be able to fold it such that just A and D are showing. And here in the beginning, that's really what we're concerned about. We really just want A and D to fold. And if it works, it kind of looks like this. All right. So we're going to practice what we know about DNA so far, and eventually we'll either have a real quiz or a practice quiz, depending on if there's no or too many Miss Clausen absences. So here we have the 2 sides we're going to focus on. If we look at the directions, again, it says we're going to label polymer A and D, the original strands. So here at the top, where it says A and D, I'm going to actually, with a pen, put the word original strand on both. Original strand, original strand.